Uh, Hello, everybody. I'm Steve Benwart. I'm the director of the Global Food and Environment Institute at the University of Leeds. You can gather from my accent, I'm from the USA. I have an undergraduate degree and a master's degree in environmental engineering from the University of Iowa. I did a PhD in natural environmental sciences at the Swiss Federal Institute of Technology. I work on soil and water resources, particularly related to agriculture. My core science is chemistry. It's a great pleasure to welcome you to the 5th virtual seminar of the Global Food and Environment Institute. The title of today's talk is People's Acceptance of Food Biotechnology in the UK and Switzerland, Gene Editing versus Gene Modification, which will be presented by Dr. Gulbanu Kaptan. The Global Food and Environment Institute is an interdisciplinary research community which brings together members from across academia, industry, and public policy to work on the integrated challenges of food security and environmental change. For further information, please visit the Global Food and Environment website. Our seminar is a part of a series of seminars, so please check the chat function with the link to our series advertised on the Eventbrite website, and look at our Twitter feed for more information. Before I introduce today's speaker, I would like to remind you of the seminar etiquette. Please ensure that your cameras and microphones are switched off for the duration of the seminar. The presentation will take about 20 minutes, during which please use the chat function addressed to everyone with your questions. At the end of the presentation, there will be a 10-minute Q&A session during which I will collate questions from the chat function. And I'd like to remind you that the seminar is being recorded and a recording will be made available on the Institute's YouTube channel. So now I would like to introduce Dr. Gulbanu Kaptan. Dr. Kaptan is an associate professor in behavioral decision making at the Center for Decision Research at Leeds University Business School. Gulbanu's research focuses on judgment and decision making in the areas of innovative food technologies, food safety, food waste, and healthy eating. She is particularly interested in research projects that require interdisciplinary collaboration between social and natural sciences. She has been involved in a number of UK and internationally funded projects, including those supported by ESRC, BBSRC, the German Research Foundation, and the EU Framework 7 program. Welcome, Gulbanu, and I'll pass the floor to you.
Thank you very much, Steve. Thank you. Hello, everyone. It's a pleasure to give a talk in this seminar series. My talk today will be on an experimental study that I conducted with two colleagues from Switzerland, Dr. Angela Berg from ETH Zurich and Dr. Sabrina Kessler from University of Zurich. I think Angela is in the audience today. Hi, Angela, and she's a great colleague to work with, really. And the motivation for this research was our interest in people's perceptions and acceptance of novel food technologies. There were quite a lot of research on genetic modification, and those studies mostly did not show real public support for genetic modification. However, there is much less research on gene editing because it's a newer technology than genetic modification, and it's more advanced. And there weren't much papers, but there were indications that people may perceive gene editing differently than genetic modification. Therefore, we decided to conduct a study to understand and compare the acceptance of the two applications. But before I start talking about our study, I would like to give some background information and briefly explain the differences between genetic modification and gene editing, so as we are all on the same page. This is the figure I found quite clear to understand in the past, so I would like to share this one with you today. Sorry if this is a very simple figure for some of you, and if you are already aware of these. But as I said, I want everyone to be on the same page when I present our study. That's why I wanted to talk a bit about this figure. But I now noticed that I didn't put the source, so I will edit to the slides later. So if you would, when you see the presentation later, you will have the source as well. The first picture is about genetic modification. So to create genetically modified crops, here there's a plant example. Scientists remove the preferred gene from one organism. It could be anything. It shouldn't be a crop. It could be an animal, yeast, whatever. They remove this gene from one organism, which is shown in red here, and randomly introduce it to another organism, here the plant. A well-known genetically modified type of crop is BT corn, for example, as an example, where a bacterial gene that produces insecticidal toxins was introduced into the part of the plant. So they remove a bacterial gene that is producing some toxins for the insects to the plant. So as soon as the insect eats the plant, it is toxicated. The insect is toxicated. So the plant is preserved. In the case of gene editing, this is a small controlled tweak to a living organism's existing DNA, rather than the introduction of a new foreign gene. And in gene editing, it is nearly impossible to detect whether an organism's DNA has been edited or not, because the changes are indistinguishable from naturally occurring mutations. So if you do gene editing, then it is nearly impossible to detect. And it is one of the concerns, this is one of the concerns around gene editing. The advantage of gene editing over traditional breeding methods is that the changes it makes are more targeted and efficient. It's quite rapid. So we expect similar changes, or we can have similar changes with traditional breeding methods, but we need to wait more, and these are more random. However, with gene editing, they are more targeted and efficient. Now I would like to give a background about genetic modification and gene editing related research and policy as an introduction to our study. After talking about our understanding of genetic modification and gene editing. Producing safe and high quality food while simultaneously handling consumer aversion towards novel technologies are among the challenges the food industry faces today. So they need to produce safe and high quality food, but at the same time, there is consumer aversion about novel technologies. So it's a challenge for the food industry. Food biotechnology applications, particularly gene technologies, are under a significant amount of scrutiny and skepticism. On the other hand, there are limited existing data suggesting that editing of genes to be under less public scrutiny than genetic modification. So the results in terms of gene editing seems more promising or encouraging compared to genetic modification. The existing EU legislation requires that all gene-edited organisms are classified as GMOs, irrespective of whether they could be produced by traditional breeding methods, as I explained in the previous slide. And this was confirmed by a Court of Justice of the European Union in 2018. So it was confirmed. There was a big debate about it in 2018, but it was confirmed. So gene editing organisms are classified as GMOs. As a result, the UK was also, before Brexit, the UK was following the EU legislation about GMOs until the end of the transition period. Although Switzerland is not an EU country, they are following the EU legislation as well. However, the UK and Switzerland are faced with the decision whether to change the regulation regarding gene editing applications. For these decisions, it is vital to take the public's views of food biotechnology associated risks and benefits. So if as a government you would like to change the policy, you would like to change the regulation, of course they need to consult to the public. As a result, DEFRA recently conducted a public consultation about the regulation of genetic technologies with a particular focus on gene editing as a means to tackle agricultural challenges in the UK. It was closed a week ago on 17th of March, and DEFRA will provide a response in 3 months. So according to the consultation document, the link is on the slide if you may want to see. According to the consultation document of DEFRA, gene editing is a technology that would benefit the UK in terms of providing abundant and healthy food with reduced environmental impact. This is the main point they make in the consultation document. And in addition, the view is that organisms produced by gene editing should not be regulated as GMO, genetically modified organisms, if they could have been produced by traditional breeding methods. As a result of the consultation, DEFRA may seek to amend the existing definition of a GMO as it applies to England only. This would mean that GM legislation would no longer apply to organisms produced by gene editing and other genetic technologies. But this will be done if this could have been produced by traditional breeding techniques. So gene editing will not be regulated as GMOs if they could have been produced by traditional breeding methods. So it doesn't apply to inserting foreign genes, etc. My talk is focused a bit on UK, of course, because I'm presenting the study here now in Leeds. So, and there's a debate now, it's a hot topic in the UK. So the focus of the topic is mostly the UK. So this study was conducted about a year before the public consultation started. However, even at that time, there was some coverage in the media indicating that UK government would approach to gene editing differently than the EU. And we conducted the study in the UK and Switzerland to investigate people's acceptance of food biotechnology with a particular focus on genetic modification versus gene editing. And when I was preparing the slides yesterday, I chose these pictures, very nice pictures from the UK and Switzerland. I think we all miss holidays and traveling freely. So sorry about that if you see them a bit irrelevant. But they made me cheer up a bit last night. In our study, we recruited participants through a market research company. They applied quota sampling based on gender, age, and education. We had 490 participants from the UK and 505 from Switzerland, and the samples were comparable in terms of age, gender, and education. We designed an online 15-minute survey consisting of 3 parts. These were people's associations and affect towards food biotechnology, an experimental part on people's acceptance of genetic modification versus gene editing applications, and individual variables such as trust and risk aversion. In the first part, participants were asked to provide maximum 3 associations. When I say association, I'm from Turkey originally. I think it's not so easy for me to pronounce association. When I say association, I mean words, images, thoughts that came to mind. So we asked them to provide maximum 3 of them that came to their minds when they heard the term food biotechnology. And then they were asked how they feel about those associations. Next, they were provided a very short text explaining food biotechnology. I have it in the next slide, and I am not going to read it, and I'm not going to ask you to read it. I just would like to emphasize that. So we asked them their words, images, or thoughts that came to their minds first. It was an open-ended question. Then we asked about their feelings about those. And then we provided this text. And what I would like to emphasize is here the focus is on more environment. Environmental benefits of food biotechnology, and GM and GE were given as examples of food biotechnology. And these are all in line with the text we provided in the experimental section. And following this text, participants were again asked about their feelings about food biotechnology. First, they provided their feelings about their associations, then they did the same after reading our text. Which is very much in line with the text we provided in the experimental part. The experimental part measured how technology, scientific uncertainty, and media format affected participant acceptance of genetic modification and gene editing. It may be better explain this part on the actual texts provided here. So for the experimental part, participants from each country were randomly split into 4 groups. They were first presented a text on genetically modified tomato with extended shelf life. It was the benefit, followed by a text on gene-edited tomato with extended shelf life. For example, there were 4 groups. Group 1 got a text on their screen, as in the picture on your left. It was like an online newspaper. They read an online. They were asked to imagine an online newspaper. So they read it as an online newspaper, and there is information about genetically modified tomato on it, and the uncertainty here was high uncertainty, I think, high certainty. Yes, scientists concludes with high certainty. So this text or the conclusions were given with high certainty. It is what we communicate to them. Group 4, for example, got a text on their screen, as in the picture on your right. Online food blog written by Sarah Brown. And a text explaining first genetic modification, then gene editing, with a phrase about scientific uncertainty, like scientists conclude with high uncertainty this time. So 4 groups. In each group, in the first group, there was high certainty, online newspaper format. In the second group, low certainty, online newspaper format. In the third group, there is high certainty, online food blog. And in the fourth group, there is high certainty, high uncertainty, and online food blog. I hope it's clear. It's not easy to explain it in one slide. Okay. And then we measured acceptance with 3 items after presenting each text. I mean the GM text, the text on GM, and GE text, gene editing text. I think I need to just skip this slide. So these are the questions we asked about acceptance, because otherwise I can't finish on time. I would like to talk about the results. Regarding participants' associations and affect about food biotechnology, 15 categories were identified through content analysis. The most prevalent of these were science and research, specific food, eating and nutrition. As you may remember, they provided associations, and they provided many associations, of course, and a content analysis was conducted. So the most prevalent ones were science and research, a specific food, eating and nutrition, genes and genetic modification. The affect ratings of these most prevalent associations were positive for science and research and for specific food, eating and nutrition. However, genes and genetic modification was associated with negative effect. The overall effect reported for the mentioned associations was near the midpoint of the scale, as you see. And this did not differ significantly between the two countries. However, after reading the introductory text, UK participants reported more positive effect than Swiss participants. So the text affected how they feel about gene editing. Regarding people's acceptance of genetically modified versus gene-edited tomato, the experimental effects on people's perceptions were assessed with a mixed model, ANOVA, with 3 independent variables: technology, uncertainty, and media. The dependent variable was acceptance. Regarding acceptance, technology exhibited the largest effect size. Participants from both countries exhibited slightly higher levels of acceptance for the gene-edited food than for genetically modified food. And the findings of the experiment suggest that people's acceptance of genetically modified versus gene-edited food is rather stable and cannot be easily changed by information provision after reading the text. To investigate participants' acceptance of genetically modified versus gene-edited food, further we conducted 2 linear regression analysis. Almost the same variables were important for the acceptance of the 2 technologies, except for gender, country, and trust. And I will explain these in the next slide. This is just to let you know about the results overall. I think it will be better with this slide. The significant variables for the acceptance of the 2 technologies were desirability, affect about food biotechnology, risk aversion, and level of uncertainty. That means the more desirable is longer shelf life of a tomato, the higher is the acceptance. The acceptance of both genetically modified and gene-edited tomato was higher if participants initially expressed more positive affects towards biotechnology. Acceptance of genetically modified and gene-edited food was lower if participants expressed high risk aversion and if participants were in the high uncertainty condition, if scientists were uncertain about the results. Gender, country, and trust in institutions were only relevant for the acceptance of genetically modified food. For these variables, acceptance of genetically modified food was higher if participants were male from the UK and had higher trust in institutions. I will skip this one as well. So I just thought that you may find the descriptive statistics about trust interesting. So I would like to just cover the discussion and implications as, I don't have time left, I think. So overall, this study shed some light on Swiss and UK residents' perceptions of food biotechnology and their acceptance of genetic modification and gene editing applications. According to our results, UK residents are more supportive of food biotechnology than people in Switzerland. And this is in line with previous findings, for example, showing that participants from the UK exhibited much higher support for GMOs than Swiss participants. A large number of participants in our study couldn't provide a single association when we asked them. They provided many different associations, suggesting a multidimensional conceptualization of food biotechnology. So it's possible that large interpersonal differences exist in people's understanding of food biotechnology, including both genetic modification and gene editing. And this suggests further research about people's understandings of biotechnology based on how multidimensional it is. And our results revealed that having tomatoes with longer shelf life is the most important predictor of UK and Swiss participants' acceptance of gene-edited tomatoes. This suggests that the importance of perceiving perceivable benefits on the acceptance of gene-edited foods. Again, prior research suggested that offering clear and tangible benefits positively impacts consumer acceptance of food innovations and technology. So communicating benefits is really important. And I'd like to thank you for listening. Hope I could give you a bit of a flavor of how public perceives gene editing, particularly in the UK. I wish we had more time to talk. After conducting this study, we decided to conduct a more comprehensive study on gene editing only. We conducted interviews with experts about their views on gene editing applications in the food domain, and we will be conducting interviews with consumers soon. So I should have more helpful results to share in 3 months' time, maybe. So I would be happy to answer your questions and or hear your comments if you have any. Many thanks.
Thank you, Gulbanu. That was fantastic. We have questions, and please, could participants please upload any further questions to the chat line, and we'll keep going. If colleagues need to leave, that's because of their schedule, that's fine too. I'll try to have go for about 10 minutes or so maximum for question and answer if the discussion keeps going. I had first question was one from me when you were discussing this, perhaps the potential of this DEFRA work of redefining for regulatory purposes what is a gene edited organism. Have any countries, are you aware of any countries that have already done such a redefinition? Yes. And if so, where and what implications are possibly being observed?
Yeah, as you can imagine, US, so they have a different definition. Okay. Japan, Australia, Argentina, these are the ones I remember. But as developed countries, United States, Japan, and Australia already have different definitions of, or this sort of definition of gene editing, and they don't apply the same regulation to gene edited foods as they apply to GMO.
Okay, and do you know in those countries, is that information in any case made available to the consumer at the point of purchase, or is that just for more food production purposes and
I think I don't know exactly, I should say, but without informing the public, particularly in a developed country, without informing the public, no regulation should be made. So I assume they communicated this.
Okay. There's another question, which is assuming gene editing is defined in the form of traditional breeding methods, how different is gene editing organisms then from biofortification, which is defined as breeding using conventional methods? Do you have a view on that?
Okay, so I'm a social scientist, of course. I don't know the technical issues much. I can just explain my view, and I'm sorry if it it's not as expected. But in traditional breeding methods, what I understand, I had an interviews with experts, so I learned from them mainly with biologists and molecular biologists. It's how they explained to me, or it's how I understood. In traditional breeding techniques, I think whatever you do is slower and more random. However, with gene editing, it is much quicker and targeted because you use CRISPR mostly, and so it's quite an advanced technique. So you know where to put the gene, or where you will edit the sequence, and so it's not random, and the process happens very quickly compared to traditional methods. But I'm not so sure whether my answer is satisfying, because I don't know much about biofortification.
Okay, good. There was a follow-up question to that, is how sure are you from the survey results that participants fully understood the difference between gene editing and genetic modification?
Well, that is why we are conducting a new study now. As I said, it was just to have a flavor. We started with an experimental study, and then decided to have a more comprehensive study, starting with interviews with experts and consumers. So, of course, there are limitations in the study, but there are indications, and the findings are in line with the literature findings, although there aren't many literature findings. So, in that sense, it encourages us.
If any participants have further questions, please upload them. I have one question final. We're through the questions that were on the chat line, Gulbanu. I would just be very interested to get your personal, just sort of some speculation about how this is potentially positioning the UK should this type of change in definition be approved, be accepted in the UK. How does this potentially change our trade position on what we might purchase from the USA, Australia, or what we might export elsewhere? Is there a sense, do you have a sense of how this can influence food supply business in the UK and retail?
Well, from what I read so far, and from what I know so far, and based on the studies we conducted, UK government is inclined to change the regulation and defining gene editing different than GMO. And the reasoning is, when you read the public consultation report of the DEFRA, is because of environmental benefits and having a better food supply, because when you apply gene editing, then you will have crops that you can grow in any season. For example, the ones I can think of now, you can do many things. So it matches the scenario we have for the future, where we don't have food for everyone. There isn't much availability, abundance of food. So with regards to that, it will contribute. Gene editing applications contribute to this. And that's why DEFRA started a public consultation. So they think that it would be beneficial for the country and for the citizens. However, before deciding it, of course, there needs to be a public consultation, and we don't know what's going to happen in 3 months, in 3-month time. On the other hand, from previous research, there are findings that, for example, there's a group of people who think that It could be genetic modification or gene editing. It doesn't matter, but it is sort of playing God or inserting new things to plants or animals. So this can't be accepted. And research shows that it is nearly impossible to change those attitudes. However, there are other research showing that because of financial reasons, which I don't expect, because I think it shouldn't be the reason to accept gene editing if you don't have, if the reason is just getting cheap food, it shouldn't be the reason. It should be more than that. However, other research is showing that people are more tolerant now about novel food technologies, and there's an advancement. And my conclusion is technology is advancing, and we know what's going to happen in the future. We don't have a very rosy scenario for the future. So there will be some technological advancement, and this could be followed. But I think here the main question is whether some companies will be advantaged from that or not. And it is one of the main question of the NGOs, rather than risks and benefits, because it seems that gene editing doesn't have much risks from what I know. However, the main question is who will produce those, which companies, would it be a set of the big companies? Then what are the farmers going to do? I think these are the questions that need to be asked, and these questions are being asked by the NGOs now.
Okay, great. We'll stop our seminar there. Thank you very much for that, and thank you for giving that more extensive answer at the end about some of the outlook for this. I hope it would be great to have you back for a seminar in a year or so to show us what else you have learned on this as it goes forward. This is a fascinating area, and of course it's extremely relevant right now. So thank you very much, Gulbanu, and thank you everyone for participating today and attending, and direct colleagues to the YouTube channel later on for colleagues that were unable to join today. Thank you very much. Goodbye.
Thank you very much. Bye.